in a space within the thylakoid. All right? So there's three different spaces. So now let's talk briefly about some of the function, okay? Some of the processes which takes place in these different spaces. Well, at least in the thylakoid and in the stroma, right? Well, first of all, when we talk about this thylakoid, this individual sacs, then their membrane is the place where electrons are transferred. Remember that when we talk about the entire photosynthesis process, when we talk about there is a need to catch those photons energy, right? Photons of light. Then to convert this energy in order to hydrolyze water and form molecular oxygen, and then to use electrons which are formed during such hydrolysis of water in order to transfer these electrons and eventually use the energy of such transfer, a certain type of other things, in order to synthesize ATP and ADPH. And all these reactions are taking place in the membrane of the thylakoid. All of these reactions, such as absorption of light by proteins attached to a certain type of pigments, inorganic compounds called chlorophylls, as well as electron transfer, as well as the synthesis of ATP and ADPH. All these things are done by several proteins and protein complexes, which all inserted into the membrane of the thylakoid, right? And this is just a depiction of such an electron transfer in a certain direction from one of the complexes or one of the photosynthetic complexes, photosystem number 2, to some other components, right? So the very first reaction in the series of reactions would be the absorption of a photon of energy. And this photon of light is absorbed by those proteins which have as their inorganic group, prosthetic group, chlorophyll, the green pigments, right? So they would absorb this energy, these individual photons. And then the energy of each of these photons would be used in order to, for this photosystem number 2, and it's just a kind of weird order. You would expect it could be called photosystem number 1, as opposed to photosynthesis number 2. No, because it discovered second after the photosystem number 1. It's called PS, photosystem number 2. All right? And so this photosystem number 2, using the energy of these photons in order to drive the chemical reaction in which the water is hydrolyzed, and the product of this hydrolysis are molecular oxygen as well as photons, as well as 2 electrons, right? And then the photosystem 2 not only hydrolyze water and form molecular oxygen and protons in the thylakoid lumen, here inside of the thylakoid, but it's also transfers these 2 electrons to the next component of electron transfer chain in the thylakoid membrane called plastoquinone, PQ, right? And I didn't write a formula here. It's a lipid soluble molecule that would take 2 electrons from photosystem 2 and transfer them to the next components of electron transfer chain, which is called cytochrome BF. This is just because the key components of this complex are cytochrome B and cytochrome F. Those very specific thylakoid chloroplast components of this complex, right? And so this complex not only taking electrons, accepting electrons from the plastoquinone, PQ, but it's also then transfers them to to PC, plastocyanine, right? Plastocyanine PC, which accepts these 2 electrons. In addition to that, the complex cytochrome BF is the only one which also out of this other component, which not only dedicated to the transfer of electrons to the next components of electron transfer chain, but also to pumping protons. Remember, that's what takes place in mitochondria as well. There are some complexes which pump protons. In this case, it's cytochrome BF. And cytochrome BF would transfer protons specifically from the stroma, the place surrounding the thylakoid, inside of the thylakoid, into the thylakoid lumen, a single proton, right? Then transfer electron to plastocyanine, another shuttling component of this electron transfer chain, transfer this pair of electrons to the next component, which is called photosystem number 1. And this photosystem number 1 by itself is not very efficient with some extra help to push electrons to the next component of this electron transfer chain paradox FD, right? But it's assisted, it's acquired some extra energy which provide an extra energy in order to drive these electrons towards the paradox by absorbing another photon. So photosystem number 1 is also surrounded by proteins which contain this chlorophyll, green pigments, right? Electrons transferred to the paradoxine, and then paradoxine has then these electrons. Finally, on the final acceptor of this electron, NADP reductase, an enzyme, which is a name implied, would reduce NADP, right? Which synthesize from NADP plus and a photon, a molecule of NADPH, the one which is required as a source of reduced equivalent for the Calvin cycle, for the synthesis of carbohydrates, right? And so this enzyme, this NADP reductase, would use these two electrons to synthesize from NADP plus and a photon, NADPH. And this NADPH was then used. And then it's very important to remember for you guys where exactly this NADPH is synthesized. It's synthesized in the stroma here, right? By an enzyme which is inserted into this thylakoid membrane by transferring electrons to NADP in the stroma. So NADPH is released in the stroma of the chloroplast. Now, as you probably remember, at least one proton has been found by cytochrome BF inside of the space within the thylakoid, into the thylakoid loop. So this proton can be returned back into the stroma, and that would release a certain amount of energy, the energy which the very same enzyme, the ATP synthase, which pump the protons from the thylakoid lumen into the stroma, it will also synthesize ATP from inorganic phosphate and ADP, and again, it will synthesize, release complex 5 in the stroma of the chloroplast, right? So now you would have NADPH and ATP both formed, one by NADP reductase, the other one ATP by ATP synthase in the stroma of the chloroplast. And then they can be used in the stroma chloroplast for this dark reaction of the photosynthesis. Okay, so the last things, let's talk about the stroma, the other space, right, the stroma. So, because these ATP molecules and NADPH molecules are synthesized, are formed in the stroma, the entire dark reaction, this Calvin cycle, when carbon dioxide from atmospheric carbon dioxide is used in order to synthesize carbohydrates using the ATP and NADPH, would take place in the stroma of the chloroplast. In addition to that, chloroplasts do have their own DNA, just like mitochondria do. Not only that, chloroplasts, just like mitochondria, are able to replicate this DNA. So therefore, inside of the chloroplast, in the stroma, there are molecules of DNA, circular molecules, many of them identical to each other, coding a certain type of chloroplast proteins. This DNA is replicated, transcribed. Those messenger RNA within the chloroplast, not leaving the chloroplast, will be translated into proteins, and these proteins are the core components of this electron transfer chain, right? Just like in mitochondria. And then, so therefore, chloroplasts do have their own DNA. All of the proteins required for replication of their very own DNA. All of the proteins which are required for the transcription, RNA polymerase, etc., of each individual gene. And all of the proteins which are required for the translation, including ribosomes, very own ribosomes assembled in the chloroplast, and so on and so on. So they kind of self-replicating machine. The thing is, very important things to remember, that it's only about several percent, 3–5% of all of the proteins of the chloroplast are encoded by the chloroplast DNA. More than 90% of all of the proteins which can be found all over in the chloroplast, in thylakoids, in the stroma, in the membranes, out there in the, they are encoded by the nuclear genes, and therefore deliver these proteins to the chloroplast following their synthesis on free ribosomes in the cytosol. Okay, guys, do you have any questions? Yeah, go ahead.
I have two questions. carbohydrates that are produced in stoma, are they the 6-carbon carbohydrates?
Yeah, it's mostly
Or
glucose.
is it? Okay.
It's mostly glucose. I mean, you know, the intermediates include 3 to 4, 5 atoms, 4, 5 atoms, carbon atoms, components. But eventually, the entire cycle is organized in a way that the product that will be then removed from the cycle is the 6 carbon, and that's a kind of drive also.
It's not like 3 carbons and then transported
No, no, it all takes place in the stroke. So it's actually a complex of proteins which conveniently assemble into the complex, which drives the entire Kelvin cycle, not just by sitting separately and then throwing at each other, you know, their product of their reaction. No, they organize the difference.
My second question is the computer side. This looks very similar to the electron transporter on the
Mm-hmm.
Did many of these pair homology with some of those?
No. Well, ATP synthase is the closest reminiscent. Well, I mean, the name is definitely the same, right? Someone would use the name chloroplast ATP synthase as opposed to mitochondrial ATP synthase. And there is another ATP synthase in the other organelles and the plasma membrane. They all much more similar to each other than, say, for instance, other components of but still, significant. There is a significant difference. Okay, guys. Any other questions? You look very new, right? You just have a few minutes left. Not really new. All right. So now let's talk today about some other organelles within the nucleus itself. None of which is surrounded by two membranes, only by a single membrane. And by the end of this lecture, it will be understandable that there is a certain explanation for that. Why is that some and not all of the organelles within the nucleus itself surrounded by two membranes? And what is the origin of these organelles surrounded by two membranes, as opposed to those other organelles surrounded by a single membrane? And so let's talk about the first pair of organelles, because they kind of interconnected, and they actually form a kind of a certain organelle system. We will use a specific name to describe this system. So the name of this organelle is the endoplasmic reticulum. Remember, the one is organelle endoplasmic reticulum, which is formed from the nuclear envelope simply by invagination of this outer membrane of the nuclear envelope into the cytosol, and it kind of creating a very complex architectural design, right? And then the other organelle we will be talking about is called the Golgi complex. And that's really a complex because if you will see, and we will go through some of the electron fluorescent photographs, some of the depiction, artistic impression about the Golgi complex, it's indeed a complex because it's composed of several organelle compounds, right? Kind of membranes surrounded structure, which do not connect to each other, are not connected to each other, and therefore, but still stay very close to each other, forming a certain type of complex, performing the same function. And what this function we will talk about. So the Golgi complex. Now, the major function of these two organelles, the endoplasmic reticulum and the Golgi complex, is to transport proteins, many different kinds of proteins, to different locations within the cell, and to sort them. Sort means that at certain points they will send a group of proteins into one direction, another group of proteins into other direction, and the third group of proteins will be sent in some different direction. So they're able to sort, especially the Golgi apparatus, some of the proteins and direct them to different places. And these places where those proteins which coming through by a certain type of flow from the endoplasmic reticulum to the Golgi, this flow of proteins and the sorting of proteins would eventually deliver these proteins to 3 different places. One of them out of the cell. Some of the proteins which coming through the endoplasmic reticulum to the Golgi, and then will eventually end up being secreted out of the cell. Say, most of the polypeptide and small protein-based hormones, they are secreted